Okay, so hi, today we'll talk about the first lecture about immunology. The main reference for the topics of the lectures will be the microbiology and immunology, the 6th edition. As well, there are other 3 references, good ones, can be used and recommended for understanding more about immunology. So for the topics of the lecture, we'll have, actually, we'll talk about 2 modules, Multisystem 1 and Multisystem 2. Each of them will have 7 different topics. For Multisystem 1, we'll have today we will have properties and overview of the immune response. Then the second topic, non-specific barriers. Then inflammation and soluble factors. Then mediators of inflammation and immunity. Then innate immunity. Then we'll talk about the antibodies. We'll talk about the properties of antibodies. Then for Multisystem 2, we'll start talking about immunogenetics, the adaptive or the acquired immunity, how body respond to antigen, and then we'll talk about the immunological aspects of cancer, and then we'll see some immunological assay to detect the antibodies in our body. Then we'll see some clinical immunological disorder divided on 2 lectures, and we conclude with the second part of the clinical immunological disorders. So actually, these are the 14 topics we will deal with in the immunology. So let's start the first topic, which is properties and overview of the immune response. So generally, the vertebrate immune response is divided into 3 axes. The first one is the non-specific external barrier. And actually, it's primary anatomical structure, such as the skin, cilia, secretions like tears and saliva and mucus. And these are actually non-specific. They cover the external surface of the body, and as well, they line the body cavities that open to the outside, like the respiratory or the digestive or the urogenital tract. So actually, they either cover the external surface, like the skin, for example, or line the internal body cavities or any tube inside your body, including the respiratory and digestive and urogenital tract. The second line of defense, actually, it's a non-specific internal defense, which is termed the innate immunity. And this actually works if the external barriers are breached. And this innate immunity respond actually non-specifically, because you majorly have 2 kinds of cells, either phagocytic cells or natural killer cells. And these kind of cells doesn't need specific behavior to fight any some kinds of pathogen. So they fight non-specifically. And you can see these actually are white blood cells, and they engulf particles non-specifically or destroy the infected cells. In this type of defense, chemicals are released by the damaged body cells, and actually this actually triggers these types of cells to do the fighting. And actually, these trigger two different and distinct mechanisms, which is inflammation and fever. And of course, we'll deal in details in the lectures about what are the difference. The third type of defense, actually, it is a more sophisticated one, which is adaptive immunity. And this adaptive immunity, or the acquired immunity, it is specific, and in which there are the body selectively destroy the specific invader, toxin or microbe. And it's not only selectively destroys this invader, but actually it remembers the invader. In instance, if it comes back to our body, it can respond to this invader faster than the initial response. So basically, it gives the memory to our body to remember this invader in future encounters with our body. And it is very specific type of defense. So let's see initially what are the terms that is related to each axis of the immune system. We will describe later in these lectures. For innate immunity, you will hear about inflammation, chemotaxis, opsonization, cell activation, and lysis of the pathogen. While in the adaptive immunity, you will hear about enhanced immune response or enhanced antibody response, enhanced T-cell response, enhanced immunological memory. Actually, this slide is like introductory. Both of these axes of the immune system will lead ultimately to the clearance of the immune system, to the clearance of the pathogen or the invader, of all sorts of invaders, through the process of tagging of this invader. So the blood-borne substance or any invader inside our blood are tagged and removed by the reticuloendothelial system, which actually filtering the blood of any invader. So basically, the host defense mechanisms actually can be majorly divided into barriers, inflammation, innate immunity, and the adaptive immunity. While there are major concerns with our immunity in case there is a malfunction of this immunity. Any disturbance in our immune system will lead to a lot of various diseases, including autoimmunity, hypersensitivity, even cancer, even susceptibility to infections, and as well immunity of our body is related to the complication to the process of tissue transplantation and the blood transfusions. So this slide actually, it's like introductory slide for the components of the immune system. So let's start as an introduction. Let's see generally what are the components of the first line of defense, non-specific one. So it involves an antigen-independent mechanism that provides the first defense against the pathogen. And as well, you can see that it's either anatomic and the physiologic barriers, and these two barriers, the anatomic and physiologic barriers, actually exclude many microbes. While the inflammation plus the vascular permeability, because inflammation had to be with the vascular permeability, and we will know why in later lectures, because this will lead to the leakage of the antibacterial serum protein. So your body generate a lot of antibacterial proteins that need to reach the site of the inflammation, and these proteins are collectively called acute phase proteins. And as well, phagocytic cells need to migrate to the inflammation site and the damaged area to fight any microbes or any invader inside your body. And of course, these two, the anatomic, physiologic, and the inflammation, all of them are non-specific. The process as well of phagocytosis, it is a process which initially triggered by the neutrophils and then later by the macrophages. And both macrophage, majorly, its main function is phagocytosis. And of course, it destroys the whole microorganism and especially bacteria. And as we said, non-specifically. And the complement system. What is complement system? Actually, complement system it's a group of proteins, and these proteins circulate and being activated in your body by the microbial surface. So it's kind of your body scan any foreign thing or any foreign invader that comes inside your body. By scanning its surface, you can decide which complement system can fight this invader, either through different pathways. We'll know later what are these pathways, either alternate lectin pathway or by immune complex through a classical pathway. And all of them are called, all these proteins that contribute in these pathways are called the complement system. So let's see, what are the anatomic and physiological barriers of our human body? Starting with the eye, you will see that the eye, of course, have the tears, and these tears, besides it wash out any microbes inside your inside your eye, it have lysozyme as well, and lysozyme is important for lysing a lot of bacterial walls. For the respiratory tract, it have mucus, ciliated epithelium, and it has a specific type of macrophage which is reside inside our respiratory system, which is called the alveolar macrophage. And we'll know later what are the function of the mucus and ciliated epithelium. And then we have the genitourinary tract, and of course you have the urine, which wash out a lot of bacteria as well. The acidity of urine as well prevent the growth of bacteria. You have the lysozyme as well, and you have vaginal lactic acid, which provide a low pH that kill a lot of bacteria. While the skin considered to be as well first line of defense, actually it is anatomic barrier, not a physiologic barrier, and it is skin can secrete a lot of antimicrobial secretions. For the digestive tract, you have the stomach acidity, which kills a lot of bacteria with ingested food. And you have a normal flora. What's a normal flora? It's a collection of bacteria that normally lives inside your gut. And you have the bile as well. And all of these are physiological barriers. For the specific immune system, so you can see the properties. The specificity is determined by the antigen. So antigen specificity, this can your body or your immune system can differentiate between any very subtle or minor changes or even molecular difference among molecules. So it can differentiate between two invaders. And as well, our immune system have a diversity. So it's not only can differentiate between different invaders, but it can generate a lot of defense molecules to recognize and respond to a huge number of different antigens. So antigen is the invaders inside our body. So as we said, we have specificity, which is determined by the antigen itself. We have diversity, and we have as well memory. So your immune system recognizes specifically the antigen. It can generate a lot of vast different number of defense molecules against this antigen, and it can remember this antigen for any further encounters and for more efficient secondary response later in years after the first encounter of the antigen. One of the major important characteristics of our immune system is the recognition between self and non-self. Otherwise, problems will occur. If there is a problem with recognition of the immune system to our body and differentiate between self and non-self, an autoimmune will arise. So our immune system have a tolerance against our self-antigen, but only responded to foreign antigens. One of the major properties as well of our specific immune system is the clonal expansion. So your immune system can proliferate very fast and differentiate very fast to resolve or control infections. What are the functional branches of our immune system? You have two major functional branches. The cell-mediated branch, which we'll call it CMI response, and these are affected by a type of lymphocytes or white blood cells, which is called T lymphocytes. where the humoral branch or humoral immune response is affected by antibodies. And these antibodies actually, initially, they are expressed on the surface of another specific type of immune cells, which is the B lymphocyte. And they can be as well secreted by these B lymphocytes from a cell that it is terminally differentiated from the B cells, which is called the plasma cells. So initially you have a B cells that differentiate into plasma cells, and these plasma cells are able to secrete the antibody, which is specific to the antigen. So we can see here just illustration of the some of the components or the major components of the innate versus the adaptive. So you can see here for the innate immunity, you have the phagocytes, you have the natural killer cells, you have a lot of blood components, including the complement system, that all participate in defending against any infectious agents. And as well, in adaptive immunity, you have two major types, as we said, the T cells, and then you have the B cells. And you can see the B cells, it have this molecule on its surface, which looks like a Y. This is the antibody, and it can secrete a lot of antibody specific to the invader when this B cells is differentiated into a very specific type of cells called plasma cells. And of course, you can see that the adaptive, the specific, the acquired immunity can last for days, weeks, months, or even years. So what are the primary organs for the immune system? You have two major organs that controlling all the immune system. You have the thymus and the bone marrow. The thymus, actually, it is the site of the maturation of T cells. And that's why it comes T cells, T from the thymus. While the bone marrow, it is the site of maturation of B cells. And that's why B for bone marrow, okay? And this bone marrow, in fetus, actually, it is the liver of the fetus carry the function of the bone marrow until it's born. Keep in mind, the major difference between B cells and T cells, actually the B cells attack invaders which is outside the cells, actually which are in the stream of the blood, for example, while T cells attacks infected cells, either virus-infected cells, either cancer cells. Okay, so that's a major difference between B cells and T cells. That's why B cells secrete antibodies against invaders, while T cells goes by itself to attack the infected cells. And we'll see that in the coming lecture, there's a lot of different types of T cells. For the secondary organs of the immune system, you have lymph nodes. A lot of lymph nodes are scattered around your body. And they actually the site where the immune response is initiated. For example, you have, for that to be occurring in the lymph node, you have a specific type of cells which is called dendritic cells. And once there is dendritic cells meets an antigen from the peripheral circulation, they enter the medulla of the lymph node where T cells stays there. The B cells as well can wait in the follicle of the lymph node for T cell activation. So the first info here, we need to know that B cells to work, it needs, or B cells to work and start secreting antibodies, it needs the activation of the T cells. While the T cells to work, it needs the activation of the dendritic cells with the antigen upon when it recognizes. Once the B cells are stimulated by the antigen in the germinal center within the follicle, they are stimulated and start secreting antibodies. The secondary organ is the spleen. And spleen as well, it is a site of immune response to antigen in the blood. And as well, its main function, the spleen, is filtering the blood from dead erythrocytes and microbial particulates, and a lot of encapsulated bacteria. The third, secondary organ is MALT, which is mucosa-associated lymphoid tissue. And actually, it exists in the intestine as a gut-associated lymphoid tissue. You can see it as well. Pear patch, it's a kind of like a mini lymph nodes. You can see that M cells, which exist in the mucosa, and they are actually a doorkeeper for Pear patch. And you have as well intraepithelial lymphocytes, so it's a kind of lymphocyte that is specific for the location, for the gut-associated lymphoid tissue, and the batrol, the mucosal lining, because as you know, the mucosal lining is the first layer that food in our gut got exposed. So if it's this food, if it's having a lot of bacteria, you will have this kind of lymphocyte, intraepithelial lymphocyte, that is patrolling the mucosal lining for searching for invaders. And then you have the tonsils and the adenoids, which are actually the glands in our mouth, and they are populated by a lot of B cells. So let's see here, majorly now, what is the difference between the innate and the adaptive immunity? Concerning specificity, the innate actually for pathogen-associated molecular patterns, which is called PAMPs. And we'll understand later what are the PAMPs, okay? That's a signature for any pathogen, either liposaccharides. It's a lot of different molecules that triggers the innate immune system. While for the adaptive, it is a specific branch of the immune system for microbial and non-microbial agents. What about diversity? Diversity for the innate is limited. But as we said, for the adaptive, it's very high diversity. Of course, the memory, there is no memory for the innate, while the adaptive, it's characterized by memory. For the self-reactivity, both of them, actually, the innate and the adaptive, of course, need to discriminate or differentiate between self and non-self. Otherwise, autoimmune disease will arise. For the components, for the anatomic and physiological barriers of the innate, you can see skin, mucosa, normal flora, even temperature of our body, pH, antimicrobials, and cytokines. While the adaptive, as we said, it's a lymph node, spleen, MALT, okay? What about the protein that participate in these two different branches? You have the complement system, as if you remember, the complement system are a group of protein that are activated by three different pathways, while for the adaptive, we have the antibodies. What about the cells participating in the two different branches? We have two major cells participate in the innate immune response, which are the phagocytes and the natural killer cells. While for the adaptive, the two major cells are the B cells or the B lymphocytes and the T lymphocytes. So let's see here how the immune system, both of these two immune branches of the immune system, the innate and the acquired, are related to each other, or in other words, who starts first for a specific antigen. So you can see here from this graph, you have on the x-axis the duration of the infection, while you have on the y-axis the level of the microorganism, the level, how much or how many of this microorganism inside your body. It starts, you can see that it starts here, the entry of the microorganism, and it starts of course, for example, the number of microorganisms increase in your body, and this will trigger the innate immune response, as you can see from the top of the chart. This will trigger innate immune response, but this innate immune response will reach its maximum if the antigen threshold level reaches a level that cannot be defended by the innate immune response. Once this microorganism reaches this level, it will trigger the activation of the adaptive immune response. And you can see that the adaptive immune response start to kick off and start to increase based on the level of the microorganism. Once it reaches the adaptive immune response, reaches its peak, the level of the microorganism will start decreasing. As you can see, it just starts first with the induction of the adaptive response. Once it becomes maximum the adaptive response, you can see that the level of the microorganism will decrease until reaching a pathogen clearance, or all the microorganisms have been cleared from your body. Not only stays here, but the adaptive immune response will provide immunological memories that can last for months or even years for the same antigen. It's only for the same antigen. So from this chart, you can see how the innate and adaptive are related for just one antigen. So for each antigen or each pathogen or each invader, you have this kind of chart. Thank you.
Yeah, thank you.